Okay, so picking back up, so this is video 3. If you have watched now, that since we have more than just a first one and a second one, so we'd finished the Krebs cycle, and so that takes us into what's going to be step 3 and the electron transport chain. But before we do that, so at the end of glycolysis or the Krebs cycle, we've only gained 4 ATP. We made 4 in glycolysis, but remember we used 2, so we only got 2, and then in the Krebs cycle we got 2 plus 2, 4. But we also got 10 NADH and 2 FADH2 from glycolysis in the Krebs cycle. So it's going to be the breakdown of these compounds that are going to give us the 34 remaining ATP to get to our total of 38 for aerobic respiration. So before we get started with the electron transport chain, just note, since we're talking about NADH and FADH2, FADH2 is broken down similarly to what you're going to see shown for NADH, but we're only going to talk about NADH just for the sake of not confusing anybody. So, NADH is broken down into a molecule of NAD+, a hydrogen proton, which is just the proton off of a hydrogen atom, and 2 electrons, which I'm going to use 2, and then an E, and then a minus sign for that as we go. So the NAD plus can be used in future glycolysis in the Krebs cycle, so that's just going to go off and find some new glycolysis happening and be used in there. The hydrogen protons are going to accumulate within the mitochondria matrix. Now I'm going to talk about what that is in a second. So they're just going to build up in there, more and more and more of those hydrogen protons, H+. And then the 2 electrons go through several what we're going to call transition molecules, and with each molecule they lose energy. Eventually, the 2 electrons join 2 hydrogen protons and an oxygen atom to form water. So 2 hydrogen protons, 2 electrons give us 2 hydrogens and an oxygen, water. Water was one of the things that even going back to 7th grade you knew was a byproduct of cell respiration. Anyway, so the lost energy from those electrons wasn't really lost. The energy is used to pump those hydrogen protons from the mitochondrial matrix into the inner mitochondrial space. And eventually we're going to have too much of those hydrogen protons being pumped out. We're going to lose equilibrium, and they're going to want to get back to equilibrium and get back into the matrix. So before I finish this off, let's look at these pictures. So here's my mitochondria. It's terrible drawing, but it's getting the job done. So this whole thing's my mitochondria. This is just the outer membrane of the mitochondria. This area in here is the inner mitochondrial space, or in the video you guys are going to watch that's in the notes packet, he calls it the outer compartment. I'm fine with either. This inner membrane is called the cristae. And then the area innermost on the mitochondria, that's what we were calling the mitochondria matrix. So it's just the inside part inside the cristae membrane and the outer membrane. So this box then, I blew up to this size so that you can see more detail. So this is the matrix, so that was the part that was in there. This is the cristae. You can see some of these proteins on the cristae now. And then this is that outer membrane. This is the inner mitochondrial space. So this part was here, this membrane there. So we're going to come back to this in a second. Anyways, so we lost equilibrium. The hydrogen protons can only re-enter the mitochondrial matrix through a protein called ATP synthase, which is this guy. As those hydrogen protons move through that protein, that ATP synthase protein, the quote-unquote flow of those hydrogen protons causes the axial part of the ATP synthase to spin. This spinning is what forces ADPs and phosphate groups together to form ATP. So that's physically how the ATP is made. So what that's going to look like. So we have a bunch of hydrogen protons in here. And so with the energy lost from those electrons, they're going to get pumped through these carrier proteins into the inner mitochondrial space. And after a while, we're going to pump a whole bunch of them. And eventually, we're going to have way more of them out here than we do in the matrix. There's only a couple in here. There's a bunch out here. So they want to regain equilibrium. We talked about transport earlier, things like diffusion. We want the same amount on this side of that cristae membrane as this side. They can't go back through here. They can't fit through the membrane. So the only way that they can go back in is through that ATP synthase protein. So as those hydrogen protons stream through there, and you guys can kind of see this part I'm going to color red, think that this is like an axle. And this thing physically, like actually rotates. It has a spinning direction to it. So the flow of these hydrogen protons going through there forces this thing to spin. And it's that spinning, and if you guys watch that video, he talks about this too. We don't actually know how it makes the ADPs and phosphates combine, but it happens within this ATP synthase protein. And again, think about that this is a tiny thing on the inner membrane of a mitochondria, which you can have hundreds of mitochondria in one cell. You are made up of billions of cells, so therefore you'd have like hundreds of billions of these little tiny proteins in your body that are spinning all the time, which is kind of a crazy thing to think about. So anyways, if you remember, we had 10 NADH and 2 FADH2 from the Krebs cycle and glycolysis. Each NADH gives us enough hydrogen proton flow through ATP synthase to make 3 ATP. The FADH2s don't make as much. They only give us 2 ATPs. So we had 10 NADH and 2 FADH2. Those 10 NADH through the electron transport chain can indirectly give us 30 ATP. The 2 FADH2 indirectly give us 4 ATP. So that is 30 plus 4, 34 ATP from the electron transport chain. We had 2 ATP from the Krebs cycle. We had 2 ATP that were actually gained in glycolysis, and that gets us to our magical 38 ATP in aerobic respiration. Like I said in the first video, or maybe it was the second video, make sure you're still looking at the notes. There's going to be some vocab terms here and there, but it's not anything that I'd probably need to go through and explain. If you folks can watch these couple of videos and have everything make sense, I think you'll be all set for the quiz. If you have any questions, feel free to send me a message. Thanks.